Hi, I'm Dr. Stephanie Duguez and working in Northern Ireland Center for Stratified Medicine. In this center, I'm leading the research group in neuromuscular health. I would like to thank the organizer to give me this opportunity to talk about our research and to talk about also our project to develop a collaborative network for neuromuscular and neurodegenerative science in Northern Ireland. Neuromuscular disorders will affect this system, this neuromuscular system. It can affect either the nerves, here, or the muscle, or the communication between muscle and nerves. Neuromuscular disorders are composed of many different diseases, and each individual disease are quite rare. But when we group them all together, they are quite significant. Indeed, in Ireland in 2010, there were 118 people being affected by neuromuscular disorders out of 100,000. Neuromuscular disorders group over 70 rare diseases altogether, and we can try to organize them according to their phenotype. And we can observe here a list of different subgroups, like the myopathies, the motor neuron disorders, peripheral nerve disorders, neuromuscular junction disorders, and cerebral disorders. All those different groups of disease have different heterogeneous phenotype, different age of onset. They can start from birth, or some of the disease will have an onset at the teenagehood, adulthood, or much later, and can have an onset in the later age here. In our research group, we are currently mainly focusing on the motor neuron disease, motor neuron disorders. So this topic will mainly focus on this subgroup of disease. Amyotrophic lateral sclerosis, or ALS, is the most frequent of the motor neuron disease. Most of the cases are sporadic, between 90 to 85% of them, meaning there's only 10 to 15% of the cases that are familial. It mainly affects male, and most of the time it has an adult onset. And once the first symptoms appear, the life expectancy is between 2 to 5 years. ALS, as I said, is a motor disease, affecting both the upper and lower motor neurons, which you have here highlighted in pink. Because of cells are specifically dying, they are leading to the muscle degenerations and muscle atrophy. ALS is an heterogeneous disease. It can have various age of onset, can start at juvenile onset, young onset, or most frequently, most ALS cases will have an onset in the adulthood, elderlyhood. In ALS, we can also observe a different site of onset of the symptom. Most of the patients will have spinal onset, whereas another subgroup of patients will have a bulbar or respiratory onset. When they experience a bulbar or respiratory onset, the life expectancy will be quite short in those patients. Today, it is difficult to diagnose ALS patients. Indeed, we don't have any kit that can confirm quickly that the patient is affected by ALS. So it means that the patients have to go through a full battery of clinical and neurological exam. And it means that they will have to go through the MRI, through EMGs, through nerve conduction velocity, and all this will take time to reach a final diagnostic. This means that there is a delay to diagnose an ALS patient, and in average, it takes over a year, 15 months, to complete the diagnostic. Knowing that the life expectancy of those patients is quite short, it means that we have an emergency to shorten this delay to diagnose those patients. So it means that today we need biomarkers to be able to identify quickly the ALS patients, and also we need to identify therapeutic strategy, as today we have no treatments that are efficient for ALS. To be able to identify biomarkers and therapeutic strategy for ALS patients, we need to understand a bit better the mechanism involved in the death of the motor neurons. And to do that, we can dig the genetics profile of those ALS patients. And by doing so, today, we have been able to identify over 30 genes to be involved in ALS, to be associated with ALS. And with these different genes, we try to understand the mechanism behind the death of the motor neurons and trying to understand the age of onset, the site of onset, and the different types of motor neurons impairments. In our group, as I told you, we are working on neuromuscular disorders. We are focusing much more on the muscle side indeed. And when we observed what was going on in ALS patients, we could observe that the muscle was getting denervated quite early, at early stage of the disease. That's what you have here on the cartoon on the right. You have here a nerve cell, a neurons, an axon coming, and the red, which is the neuromuscular junction, so the connection between the muscle and nerve here. That's what you have in an healthy muscles. In ALS patients, the neuromuscular junctions are denervated or partially denervated. That's what you have here, and you can see the axon here, the axon becoming atrophic, smaller and smaller, like if it was dying from the periphery toward the center of the central nervous system. So when we observed that in the muscle vesicle patients and also in urine model, we were starting to wonder if we have a problem in communication between the muscle and nerve in ALS patients. What if the muscle is not communicating anymore with the motor neurons, and that's why the motor neurons are dying? Or what if the muscle is secreting toxic elements that will be responsible for the death of the motor neurons? So we wanted to dig into this mechanism, trying to understand how the muscle can communicate with its environment here. And to do that, we did launch a study on muscle biopsy done on patients, and we extracting the muscle stem cells, that's what you have here in green, that are present in all muscles. That's what you have also yourself in your muscles. Those cells stay quiescent in your body. When you're healthy, there's no problem. They stay here just when you need them, when you need to repair your muscle. And what we did extract those cells, and we compare the profile, how they behave, ALS versus healthy subjects. I'm not going to go into detail here, but it's just to let you know that by taking those cells from patients, we have been able to show that those cells from ALS patients are secreting vesicule. That's what you have here on the top of the cartoon. You see those bubbles here? They are secreting by ALS muscle cells. You also have some in healthy, but in a lower quantity. And when we take those vesicles and we apply them in motor neurons, so we can have in a petri dish human motor neurons that we can generate there, and we add those vesicles on those motor neurons, what you have here in orange, a nice motor neurons here and here. And you can see that when you have ALS vesicule, the motor neurons are shorter, less ramified compared to the healthy motor neurons. That's what you have here on the right. And this is a quantification we have here. When you add ALS vesicule to those neuron cells, we are killing them. This is the percentage of death here. When you have healthy vesicles, we are protecting the neurons. In other words, the take-home message is that the ALS muscle cells are secreting toxic vesicule. So based on those data, we can then dig the content of those vesicule, those bubbles. That's what you have here. You have the exosome, the vesicule, and those vesicles can contain proteins, RNA, or lipids. And by studying the profile of the ALS vesicule, we could then identify the toxic elements inside those vesicles and probably target those toxic elements to correct the toxicity of those vesicles. So based on all this research done in the lab in a petri dish, we can open up the research here, try to study those vesicles. That's what we have done, we study their contents. By studying their contents, we know that those vesicles can go in the body fluid, in the serum, for instance, in the blood of the patients. So we can take the blood from the patients and then extract those vesicles from the blood and try to see if they contain ALS vesicule there. So this could be used as a potential biomarker. In addition, as I said, we know that those vesicles are toxic, so we know that we could try to correct this toxicity by either blocking the uptake of those vesicles or by changing their contents. So this project opened to two routes of therapeutic for the patients, for biomarkers, and for the therapeutic. So in summary, regarding our research on ALS, we did observe that ALS muscle cells can secrete neurotoxic vesicles, that the muscle vesicule and their content can be used as a target for therapeutic, and that the ALS muscle vesicles, we know that they can be extracted from blood samples. So meaning that we could use them as a biomarker for ALS diagnostic and prognostic. So in summary, we have been able to develop a strategy to extract muscle vesicule from body fluid in ALS patients. And this allows to track the muscle health status in the ALS patients. But it means that also that we can use this strategy in other type of conditions to track the muscle health that we could do in aging, that we could do in different types of neuromuscular disorders, or when people are practicing exercise or not, or in different conditions such as obesity. When we started to build our research group here on neuromuscular diseases, we discovered that there were several regional needs. Of course, there is a need for better diagnosis and for better treatments, but this is not specifically specific to the region, but still needed, obviously. But when we talk to patient group, it seemed that they needed to hear a bit more what was done on the research to address their conditions. And when we talked to clinicians, we realized that there was not enough clinicians dedicated to this field here. So we started to build our network to try to fill up this gap. And we have been successful in recruiting Gavin, who is a medical doctor, registrar, doing his PhD with us. So we have been funded for him to do his PhD with us by the ABN Association and IICN. And thanks to that, we have thus a medical doctor that will make the link between the researcher and the neurologist at the hospital in Belfast and in Derry as well. And thanks to that, we can start to develop our first Northern Irish biobank for motor neuron disease. And for this biobank, we are not now allowed to recruit patients. We have been accredited the consent for that. We would like to recruit different volunteers to give samples to our biobank. Of course, patients affected by motor neuron disease, ALS, and other motor neuron disease as well. And also healthy subjects. We need healthy subjects because we need to be able to compare the conditions from motor neuron disease patients against the healthy to see what is normal and not normal here. So we are recruiting patients for that, volunteer, and volunteer can decide whatever they want to give us. It could be blood samples, urine samples, muscle biopsies, cerebral fluid, or skin biopsies. And if anyone is interested to participate to give samples for this biobank, they can contact us, either me or Gavin. And be aware that this study is sponsored by the Ulster University. The local development of this NMD biobank is the first milestone of the development of our network, collaborative network on neuromuscular and neurodegenerative science. In this network, we are developing our collaboration with researchers working on motor neuron disease, but also on different types of neuromuscular disorders, such as Duchenne. And we are also working closely with neurologists, including Gavin and Dr. Kuladnunagi. And we also wish to work closely with the patients' associations to help us to transfer the research done at the bench and transfer it toward the patients. And for that, we are working closely with the Northern Ireland Rare Disease Partnership Associations, as well as the MNDA Association Northern Ireland. I would like to thank our research team here, So you can see that we are specialized in two types of research approach. We are a group of biologists here and a group of bioinformaticians. And we try to do transversal research and to try to communicate between the two arms of research we can develop in our group there. And I would like to thank our different funders that support our research here and our collaborative network, like Dr. Dumonceau at UCL, as well as Professor Andrea Malaspina, Dr. Cécile Martina at Einstein in Évry, and César Knoblar working in CNMC in Washington, and Helen Blasco at Tours University, and Cédric Raoul at the Neuroscience Institute in Montpellier, as well as Professor Pierre-François Prada, who is working at Pitié-Salpêtrière, also being a visiting professor at Houston University. And thanks to his all collaborative network of clinicians, we have been able to collect all the samples that we have been able to develop the study on that I've been present for you today. Thank you for attention, and I'm open to any questions. Thank you.